Hi everyone, my name's Carol Treasure and I'm the Chair of Trustees at Frame. I've been working in the field of toxicology for about 30 years now, since I did my PhD in the Frame lab.
I'm delighted to introduce Julia Fenton from SEAC at Unilever to give our presentation this evening. Julia is also an alumni of Frame, having done her PhD in the Frame lab at Nottingham and worked as a scientific officer at Frame for a few years. She then went to the European Reference Laboratory in Italy, where she was involved with validating some of the very first alternative methods. Julia has now been with Unilever for 24 years and has been instrumental in driving forward next generation risk assessment with her team of talented scientists.
It's a real pleasure to speak to you all. It has actually been a very busy year when it comes to thinking about chemicals testing and the use of non-animal approaches. And, you know, I did actually give a Frame lecture in 2005, and I took a 15-year period and talked quite personally around the successes and the frustrations of the period from 1990 to 2005. But I thought, you know, it's November and we're living with a lot of bad stuff at the moment. And it'll be dark and it'll be raining, and it was, yeah? So, you know, we don't really want to talk about the missed opportunities of the last 15 years of REACH. What I do want to do is try to give us some hope that actually we are at a tipping point, and that the advanced science and technology that many people across the world have been working on hard, passionately over the last 20 years or so is actually going to make a difference, and it's going to make a difference over the next few years. We have a few years to really grasp this and to really push it forward. So that's what I'm going to talk about. And first up then is just to thank the fantastic team at Unilever and with lots and lots of collaborators across the globe that we work with. Science is a team sport. And I'm really pleased also to say that I have the support of some of them here tonight. So Gavin, Carl, Renato and Gemma are with us tonight, which might make it easier if we have some difficult questions later on. Yeah. So in Unilever, our approach to product development and innovation is very much based on the principle of safe and sustainable by design. And we changed our animal testing policy in 2018 to reflect consumers' concerns and to embed our non-animal safety science. So we felt that our science had got to a point in mid-2018 that actually we no longer needed to rely on animal testing to assure the safety of the ingredients and products that Unilever makes. And I'm going to scope it like that because Unilever is a consumer goods company. It makes household products, cosmetics, and foods. It is not in the pharma territory because I think some of the challenges are quite different in the different sectors. But of course, we had, as Unilever, lived through the cosmetics testing ban. Yeah, and I think that is quite important in terms of how research was incentivized and how we took some of those non-animal methods into safety decision making. So I'll try to talk a little bit about that as we go through. So I've structured the talk around three areas, really. I'm a scientist. I'm a toxicologist, biochemical toxicologist by training. So I'll start with the safety science. That's where many of us are quite passionate in terms of the new assays and tools that have become available to us actually since the human genome was sequenced. So that was sequenced finished 2003. We've had a lot of things come online since then. But I will try to put it in the context of the chemicals regulations, particularly because, well, in a EU situation, the chemicals regulation, so the REACH regulation, is being revised. And also in the UK, we're trying to transpose that into UK legislation. And then a bit of backdrop around the chemicals policy. So just trying to give that broader context in terms of where the science fits in and what's blocking the science and what could be some of the unlocks. So Carol sort of mentioned we've got quite a lot of new science, and you just think about the technologies and models that are now available to us, not just from the biological or chemical side of things, but also digital technologies. So the computational modeling and some of the statistical approaches that are now used in that modeling space are way too complex for me to understand. They have changed phenomenally over the last sort of decade. We heard about systems biology probably back in 2002, 2003, but actually by the time you can map biology on a computer screen and you can interrogate that biology and what chemicals may interact with that biology and look at pathways and look at receptors and look at enzymes, that's what really changes the game when it comes to them being able to actually ensure that chemicals are safer than what we get out of animal studies. And I think there you can see, and you're probably all very familiar with, the omics technologies, microfluidics, organ on a chip. The one thing I'm just going to point out is point of departure, because I'm going to come back to how we interpret some of the non-animal data. So the point of departure is when you've essentially got your concentration response curve and all of a sudden you start to see the deviation. There's a point at which you flip from something where there's very little biological effect to where you start to see a dose response of the effect. And that's an important threshold, because as we know, biology is essentially about thresholds. And tipping over those thresholds is what we need to understand in terms of whether we're in a low-risk situation or a high-risk situation. So just back to the cosmetics bit and where the new approaches fit in. So I think, through the 1990s, certainly by the time we were getting to the mid-2000s, we had approaches that were used to assess the safety of cosmetics, but they were mainly for local acute effects, and we were sort of getting there a little bit on some of the skin sensitization, skin allergy type of methodologies. But the intractable was what happens if a chemical passes through the skin or is inhaled and actually you've got systemic exposure. And that's where I think we've still got some people thinking that we've got a block there. And we knew that we would have to be able to address that by the time we got to 2009 with a testing ban in place in the EU for cosmetics, and 2013 with the full marketing ban. So that was the incentive a little bit within Unilever way back in 2003, was what we're going to do about systemic exposure. And this is where the terminology next-gen risk assessment comes from. And you'll see in a minute that actually we published an ATLA paper with our thoughts, but the tipping point was 2007, when the US, under the National Research Council, published toxicity testing in the 21st century, a strategic view of what was required. And the US at that point started to take the leadership in this space because they put the investment behind that strategy and they translated it into annual work plans. They were quite transparent about what they were doing. They started to collaborate quite broadly beyond the US with some of the best scientists, and they started to build in the exposure science piece alongside the hazard piece. So the US, I think, from that point have really sort of pushed the agenda in this space, and they are the ones, to some extent, I think we could credit with a lot of the work that's essentially about understanding human biology, some of the computational modeling and the chemical interactions. But as that was happening in the cosmetic space, the industry and the government authorities got together under the International Collaboration for Cosmetics Regulation, and they did publish in 2018 essentially the principles for next generation risk assessment. And we saw eventually in March 2021 the EU Scientific Committee on Consumer Safety, SCCS, actually put in their notes of guidance the new approaches and the next gen risk assessment. And I heard yesterday the co-chair of the SCCS talk very passionately about those approaches and why essentially are we still doing animal testing in the chemicals area when essentially they've written that into the notes of guidance. So just so everyone's on the same page, I mean, what's different about the next generation risk assessment is more that you've got an integrated approach, which is quite multidisciplinary. And this is where I think, science being a team sport really comes in, because yes, you've got the biologists and the chemists, but increasingly people who are recruited into this area have got mathematical modelling or informatics backgrounds, bioinformatics, cheminformatics, and it's how you can actually put those teams together on some of the big challenges, I think, that starts to give us the unlock in terms of how we apply the data for decision making. We've tried to bring it to life a little bit through the people who work in the labs. So there's a couple of YouTube videos that we did with the US Society of Toxicology, just to sort of exemplify the types of tools and how we actually take the data from those tools and build risk assessments. And I'll try to cover a bit of that tonight. I think that one is just giving you really a bit of an overview of what's in the ICCR sort of approach. I think the documentation piece is increasingly important, because we need to understand the sources of the uncertainty, and we need the transparency, because one of the challenges here is always going to be about building confidence in the approaches. So the more we document very clearly the workflows, the frameworks we're using, and give case studies and examples of what we're doing, the more I think we'll try to be successful in building that confidence. And we've seen now various sort of schemes be developed for actually how the various assays and the data start to get put together in these tiered assessment and testing approaches. And there's a lot of work going on under OECD, for example, to take those forward. But this was one that came out in 2017, one of the EU research programs, which actually shows from the quite simple decision making that might be needed in a screening or a prioritization sense, right the way through to essentially being able to do a non-animal safety assessment, what is called ab initio, right from scratch, without knowing anything or any historical background to that. And so you see some common assays starting to come out. And I think from a chemicals point of view, the key thing is the physiologically based kinetic modelling. And this is why chemicals is so different from pharma or agrochemicals, because kinetic modelling has never been a part of the data requirements, but it is actually key to how you interpret the data and how you can piece together data from different assays in a decision-making context. So if I had to say, what was the big mistake probably of the past, it was that we didn't have that kinetic modelling in a chemicals context. Now, of course, companies went off and did that kinetic modelling, sometimes because they needed to refute the animal test results that they were getting, and sometimes because they knew that that was the best way that they could interpret their data. So it's not that we haven't got some of that data. Some of it was also done from an occupational exposure point of view, but it is a weakness when you start to look at the thousands of chemicals that are now used in commerce. Most of the time we haven't got good kinetic data. So our toolbox, and this isn't set in stone, it will continue to evolve as science evolves and as our experience evolves. But essentially we've got the kinetic modelling, and then we've got in vitro pharmacological profiling, we've got transcriptomics, and then we've got a whole load of cell stress pathways. That is really our basic toolbox, and you'll see how that fits into frameworks which we supplement with other assay types as we go through. But it's important that we understand that the data from those have to be integrated with actually an understanding of the exposure. That is how we actually take decisions, because ultimately all chemicals are hazardous. They have various properties. You can regulate REACH potentially on a hazard basis, but it isn't actually real life. And I think what we'll end up with very hazard-based regulations is essentially you take away some of the good chemicals, and if you're not careful, you actually leave society in a far worse place because you've not got access to some of those materials. So there's a big friction at the moment around how you build in some of the exposure piece back into REACH for decision making. And also, I think, we've got a bit of a conflict at the moment going on between the NGOs in the animal protection space and those in the environmental sort of space. Now, actually, personally, I think there's a middle ground on that, because some of these tools and techniques give us far better understanding to be able to take decisions on the safety of chemicals. So, it's one of those polarized views at the moment that actually you just want to have the proper discussions and dialogue and say, let's, take a few steps back, because actually the solution is there in front of us. It's the new science, it's the new data that we can generate, and how you put that together. Right, in 2020, the team had been busy working on a case study, and they took a chemical for the case study that I knew very well. I didn't have any say whatsoever in the chemical that was chosen, but it was coumarin, which was actually what I did my PhD on. And they started from the basis that there was no evidence whatsoever on this chemical, and how they would build up data to be able to take a decision. And you get to a framework that eventually looks like this, and I don't want to be dismissive. This was probably about 3 years worth of hard work of about, 25 people. But yes, okay, you need to understand the exposure piece, but you start to get to a point where you understand which assays are the most relevant for the data generation. So you see there, we've got the cell stress panel, we've got the high throughput transcriptomics in there, we've got something like Tox Tracker. That's about assessing DNA damage. It's got a mechanistic basis to that assay. And then what happened was they generated all of that data, and I think I could have probably predicted this. They said, we can't actually make a decision because we don't understand the metabolism. Metabolism is very important in coumarin toxicity. So they then looked at the metabolism and could start to refine the risk assessment. And eventually you get to a position where you're looking at that point of departure across all of those assays. You're looking at which are the most sensitive, you've built in your metabolism understanding, and you can start to integrate that data and make a safety decision. And then the Skinology team that had been busy beavering away for many years in Unilever, because this was the area that we did tend to lead some of the scientific understanding on. And I feel a bit bad talking about this when Gavin's in the room, because Gavin's the one who's led most of that work for the last 15 years. But they thought, okay, if the systemic tox guys can build a framework, so we can build a framework too. So this is the framework they came up with. And you can see the bit that starts to be quite different are the assays for data generation. And some of you might recognize those assays because there's some of those that are in the OECD test guidelines. HCLAC, Keratosense, DIPRA, they're all ones that have been quite widely evaluated now. In that context, though, it's still quite challenging to look at those data and think about the potency and the risk of the likelihood of having inducing skin allergy. And so they always tell me it's more complicated. We need to do more. And so, of course, they wanted the best model and the best data and lots of complicated charts. But this is a bit the journey that Gavin has been on, starting with us working with a systems biology company not long after he joined us, right the way through working with clinical and experimental dermatologists, and then grasping the modelers, the Bayesian statisticians, etc., and building what we now have, which is a Skinology risk assessment model. And all I see coming out from that team now are things that are differential equation, pages and pages of differential equation. Please, someone interpret these for me. Yes, right. But if you simplify it a little bit, where are we now? We've got that whole thing of a risk assessment framework. We've got the Skinology risk assessment model, and we're now, having published that model with all the appendices of all the equations, we're working with the US government in terms of the regulatory acceptance of that. So we're working with the US department that leads on alternative methods, and we are already talking with the OECD in terms of the approaches that they have in place. We've also published the application of that with key chemicals that are known skin allergens, so that you can see how you take that into the actual decision making around what levels of those types of chemicals that can cause skin sensitization you could safely use within products. So we've integrated all of that. And then those who are a bit more interested in the reproductive toxicity have likewise published a framework this year for development and reproductive toxicity, again with different assays, very much anchored in our biological understanding. And then we have a similar framework which we haven't had as much time to work through at the moment, so it isn't published yet. But what happens if you are exposed through inhalation? How do you assess the toxicity there? So that's where we stand with our risk assessment frameworks. Just to point out that a lot of the good research that the team has done, a lot of the understanding that we've had of how to do this has been done jointly with the US EPA. So back to 2007 and that investment that happened in the US, essentially we have been able to have collaborative research and development agreements since 2015 with the US EPA scientists. And there are 400, a mix of exposure scientists, hazard scientists, data scientists, modelers, etc., sitting in Research Triangle Park. And that is something to really sort of nurture and unlock if we're really going to drive a change in terms of how we do chemical safety. Right. Just onto quite a hot topic at the moment, particularly this year, has been, okay, so you've got your assays, you've got your frameworks, you know how to integrate the data, but you're not predicting what we would see in an animal study. Yeah? No, we're not predicting what you'd see in an animal study because actually we think we want to be predicting what might happen in humans. So this whole thing around protection versus prediction, and we talk a lot about predictive toxicology, but actually as a risk assessor, scientist, or what have you, in my job, to some extent you're used to putting safety factors on animal data if you're trying to interpret them and extrapolate them for human health risk assessment. So we would say we can do next-gen risk assessment, but it's about protecting human health. It's not about predicting, you know, which organs may actually be impacted at high levels of chemicals. But what we're finding now is everybody's sort of pitching in around the same thing, which is a new metric. So where we're used to using margin of exposure or point of departure when we've been interpreting animal data for risk assessment, we now have a bioactivity exposure ratio. And this work has been really driven by Katie Paul Friedman out of the US EPA. But I think the key point I want to land here is at no point with NGRA are we attempting to predict the results of high-dose animal studies. And this bioactivity exposure ratio, the work done by the US EPA, picked up also by Health Canada, and also trying to bring the European regulators in under an initiative that's known as APCRA, Accelerating the Pace of Chemical Risk Assessment. I think we're showing that for the majority of chemicals, so for 89% of chemicals in the US databases, then using the point of departure from the novel approach methodologies is more conservative, i.e. more protective than a traditional point of departure from animal studies. But there's going to be needed to be much more work done, much more data generated there to continue to develop the confidence in terms of how those would be applied in a regulatory context. And finally, just to finish up where we are from a Unilever point of view on the science, Alistair Middleton and the team in July published essentially what is the workflow that we're using, and also an initial prediction decision-making model that comes out of looking, I think, at 26 different exposures for 11 chemicals. So it's a pilot study, but it actually starts to pull all of this together into something that actually others could repeat. And we will carry on that evaluation. So with that model, we will take that forward and look at many different chemicals and exposures. And we've tried to do that as transparently as possible so that other people can engage and also try to test the chemicals that they're interested in, for example, in this scheme. So it's got 3 modules, and you can sort of see where the kinetics is coming together with the various toolboxes And then there's a way of actually estimating the bioactivity exposure ratio. So it does actually walk through the various steps that are required. And because we can't compare with animal data, we have to work out what we're going to have as the thing we're going after. And so that is essentially the existing safety decisions. So whilst we're using materials that have been well characterized and we understand the safety profiles, what we're looking at is that you would take the same decision on those chemicals as to whether they're low and acceptable risk or whether they're high risk from the novel approach methodologies. And the question really is, can we establish a threshold for the bioactivity exposure ratio above which we consider a chemical exposure scenario to be low risk? So that's the hypothesis to some extent that we carry on working at and trying to build the database around. So moving on for a bit different, just how this then all sort of fits together, one within a company setup, but then within a regulatory and a chemicals policy setup. So our science was at a position where in July 2018, when we had one of our biggest brands come and ask us whether they could start to make no animal testing claims, that we said actually from a scientific point of view, we feel comfortable and confident now that we can support the safety of the ingredients and the products with our new capability. And so we've linked our science into our brands, and they are starting to make no animal testing claims certified by the NGOs. And we've linked it also with our partnerships, and by partnerships broad across academia, other companies, government organisations, and I think importantly, and I would shout out this as a big change from where we were several years ago, which is partnerships based on trust and openness and transparency with some of the big NGOs. And they have very good scientists, and you can see the impact of the scientists actually on some of the papers that are coming out now. And we've all linked all of that into carrying on our work in terms of advocating for regulatory change. Now, a lot of the work we've done there since 2011 was related to China, part and parcel of a UK-China government activity around trying to remove mandatory animal testing for cosmetics products in China. And we have seen those Chinese regulations evolve. A lot of hard work by some people in the room here, and also Judy MacArthur-Clark, who's led that from an ex-UK government, but linked into the Foreign and Commonwealth Office. So we've had success there, but actually there's far, far more that needs to do in terms of that advocacy for regulatory change. We also came out in 2018, towards the end of the year, with support for a global ban on cosmetics testing. So we'd been working under the context of an EU ban, but as a multinational global company, you really want a level playing field. We did that, and at the same time invested considerably working with Humane Society International to set up a collaborative activity to help develop the capability that was needed. So to share our safety science capability, our non-animal approaches more broadly, and to involve many more players. And we've done that under the auspices of the Animal Free Safety Assessment Collaboration. So HSI have the lead on that, but that's a broader stakeholder group. The training materials have taken an age to come together, but they've started to share those through webinars. And Renato's here tonight, and he's been taking those straight into the training of the Brazilian scientists. So it's good to see that the materials, the webinars, all those types of things that have kept us busy during COVID and lockdowns are actually starting to have an impact in terms of being able to build more broadly the capability in non-animal safety science. But unfortunately, whilst we had our eyes on the rest of the world, the UK and the EU decided it was going to make life a little tricky. And life changed in August 2020, when all of a sudden the Daily Mail, just before we're breaking for the August bank holiday, get in touch and want a quote on a Friday, because there's a few UV filters, sunscreen ingredients, that ECHA are starting to ask for new animal data. So new animal testing on existing ingredients with a long history of safe use. So the focus then switches to what is ECHA doing in terms of some of its compliance checks of its dossiers, checking information that's missing, and then immediately writing letters to companies saying we want animal tests done, without any consideration of whether or not animal tests are needed, whether there are non-animal approaches that could be used to fill those data gaps. And then the UK suddenly decides that, you know, after all of the work that went on, and we had led the way on as to whether or not there's changes in how the interpretation of the regulations, what ECHA's doing, that would actually lead to cosmetic ingredient testing in the UK again. That is subject at the moment to a judicial review, so I shall not say too much on the UK position on that one. But this is the crux of the matter. The crux of the matter is that because ingredients that are used in cosmetics are chemicals and are subject to the chemicals regulations, and you see there very clearly that you've got bans on one side, and we're pushing for more bans, and yet in the chemical space, particularly those in red, you have got explicit requirements for chemicals testing in animals. So I think by this stage, the scientists were getting even more frustrated than usual, and decided that they were going to find a voice. I think those of us who had worked so hard through the 90s and had got the experience of doing safety assessments under an animal testing ban in the cosmetics area, I think it was a bit of a red rag to a bull when we saw ECHA coming with requirements for new animal tests on existing ingredients. And so we started to speak up, and increasingly, I think over the last 2 years, we've seen more and more scientists starting to speak up and say, we actually have the approaches. What is blocking us is essentially the fact that the regulations are outdated. And we've seen that, the Dove brand and Body Shop and other brands have joined in, which has helped attract more consumer attention, because I think on the whole, most consumers thought this was a done deal, no animal testing for cosmetics. And then we did take the leap, and it was a big leap for someone like Unilever to get involved in a European Citizens Initiative, and Gemma's in the room, and Gemma has led that from a Unilever and Dove perspective with the animal protection NGOs, and this is another change. We had 50 or 60 animal protection organisations across Europe all working together, all with one common aim, and Gemma and I were there when we got to the point where we got 1.4 million signatures, very hard won, with all of those NGOs. And just to see the commitment, the passion, all of the work that had gone on from the NGOs on the ground in countries through COVID, through the backdrop of a Ukraine-Russia war, all of those types of things that really get in the way of normal advocacy and activism, the creativity that some of those groups showed to actually keep this on the consumer agenda was amazing. And I'll come back to that in a minute. So in Unilever, we decided we'd write a paper, because that's what scientists do, isn't it? We write a paper, we put some thoughts down. Only not often we have a title that we're allowed to have, 3, 4, 5 lines of text, which is quite inflammatory. But actually, that was the point. Animal testing as a last resort was there in reach. Its backdrop is the 1986 Acts on Animal Protection, and yet there was no consideration to actually upholding that core principle. And then what we've done subsequently is actually calm down, and the scientists then wrote the paper, which is more around showing the science and how it can be used for regulatory purposes. But I think, from a point of view of keeping the discussion going, this whole thing around doing an animal test because it's a regulatory requirement, vis-a-vis thinking things through from a scientific point of view, and what are the tools and the data and what's the decision you're trying to make. That is the big gap at the moment that exists, I think, between the investigative scientists and the regulatory community. We were helped a little bit by some US work around laws and regulations impeding modern non-animal science uptake, which of course was important to the US agenda in relation to where the US EPA had got with their approaches and wanting to use those in chemical dossiers and for registration purposes. So what we've essentially been doing, and Carl and Gavin have led a lot of this work, is trying to translate our experience and what we've been doing for many years in the cosmetics area into the chemicals area, accepting that the chemicals area is about occupational and environmental safety. But as Carl likes to point out, you know, a human being, whether they're a consumer or whether they're a worker, the biology tends to respond in the same way. And actually, we have better safety controls in factories where you're actually aiming to minimize exposure to any of the chemicals that are being used in the factory, vis-a-vis putting it on your skin or washing your hair with it, etc. So, you know, there's something around some logic that's missing in that space. From an environmental point of view, then likewise, the environmental scientists have been working on various tiered approaches which don't require animal testing for many years, and that's becoming more prominent now. I think we're seeing more through the scientific societies like CTAC, for example, of presentation of the work that's been ongoing in that space. So I don't think there's always a need to resort to lots of fish testing either. It's the same sort of principles that need to be applied. So we've now got a few frameworks emerging in terms of how to use some of these NAMs for chemical safety, and a mix of work coming out of Europe and the US. A very good review at the bottom there, coming from the US, again, multi-stakeholder group across government and NGOs. So we also thought that the only way we're going to crack this a little bit is to make sure that we have some open dialogue with the chemical manufacturers. And so we have taken the suppliers of some of our ingredients and started to talk a little bit more about how to uphold animal testing as a last resort. And Carl's been particularly busy leading this for the last few months, where not just as Unilever, but involving some of the consultants that we use for some of our dossiers, etc. We've started to open up the dialogue around, well, what are the options? You know, if you're asked to do animal testing by ECHA, yet you've got animal testing as a last resort as a principle, what are the steps that you should be looking at? What are some of the tools that are available to you? And of course, Unilever's made itself quite popular by opting out of some of the consortia and saying we're going to do NAT, so no animal testing approaches to fill data gaps. And we're going to have some interesting discussions maybe in a few years' time, because if those are successful and yet other parts of consortia have gone on and do animal studies, we will then have the evidence around what animal testing as a last resort really does mean. So I think, you know, lots to come in that space as we go forward. And then just thinking where we are on the chemicals regulations. So I think I've sort of said the key bit that we've been focused on is the animal testing as a last resort. But I think the other bit that's going to be quite challenging for us is very much how regulations are based on hazard and don't tend to build in the exposure science that you see, for example, in the cosmetics area or in the foods area, where you do weight of evidence safety assessments. Everything comes to a head quite often if there's a company who doesn't agree that animal testing is needed, and these tend to end up at the European Court of Justice. So there's been several quite high-profile cases. I just go one from 2015, where actually the decision was that ECHA needed to ensure that companies were upholding animal testing as a last resort. Well, we're 7 years down the line, and there isn't really a mechanism or process for doing that. And then one from January last year, which was a very interesting case, and I've you know, on here, it's just the sort of narrative that came out of Advocates for Animals. But this was a case where Esso, Raffinage and others essentially were successful in their adamant defence of a weight of evidence approach that did not require new animal testing, because you could actually relate back to animal testing that had been done on similar types of materials. I think we're going to be testing the European Court of Justice again shortly. I think I'm off to Luxembourg next week. We are interveners as Unilever, along with Peter and Cruelty Free Europe, and a Simrise case where we're talking about UV filters and whether or not animal testing was needed. So we don't probably really want to keep going through this circle, but until we get to a point where I think there's more flexibility in the regulations and there's regulatory use of the NAMs, then this is what is open to us. Decisions on chemical safety, I think I've probably covered this. I probably want to just point out that Health Canada have nicely taken a lot of the work that was being done in the US and actually started to embed it in their regulatory approaches. And I think this is sort of the leading edge at the moment in terms of the work that they're doing on prioritization versus actual risk assessment, and how they have put in the bioactivity exposure ratio and looked across many types of chemicals to get to a point where they're starting to feel quite confident in the use of the NAMs. So I think, watching where Health Canada goes on this one is going to be probably the sort of the leading edge in terms of application for regulatory purposes. There's a nice case study that they have done on endocrine disruptors, which actually shows that we shouldn't just be saying and defaulting that we haven't got non-animal approaches for endocrine disruptors. I think in that situation, there are some good assays available, and it's about being creative and trying to trigger mechanistic understanding that actually informs the decision making. And on the REACH revision, which is ongoing and seems to have been delayed, probably put back for 12 months, again, the Unilever position has really just been to try to focus on the use of new approach methods, non-animal methods, and ensuring animal testing is a last resort. The shift that has happened, and I know there's a lot of hard work behind the scenes, but you've certainly seen it over the last 6 months. We've started to see ECHA be much more open to discussing NAMs and to thinking about the regulatory use and how they would sort of evolve the regulations. So where, probably 18 months ago, there was a massive gap, that gap is closing. There's some interesting minutes from the ECHA Management Board in September, which shows that they're now starting to pick up the third stream of REACH, which was to promote alternative methods, as well as ensuring a higher level of protection of human hazard and of human health and the environment. And I think, we've seen everybody sort of pitching into the same space with proposing some of the same types of solutions. So I think we're now in a position where there's agreement and commitment to a roadmap. I think yesterday, Gavin can talk about this maybe later, we heard the Commission say that they are committing to develop a roadmap to phase out animal experiments animal tests for chemicals. So watch this space on that one. UK. The UK situation, for those of us who are a bit distant from the government setup, comes across as being quite complex in this space, because there's lots of players, different responsibilities for policy versus regulatory implementation. And of course, we've got the backdrop of the ever-changing situation in terms of implementation of what was EU legislation. But I think we've got to be careful we don't miss the opportunity of the fact that the UK is very strong scientifically here, across academia, industry, NGOs, into government. We do have a lot of experience and expertise in the non-animal approaches. And so the opportunity really is to work our way through probably the complexity of the different groups and parts of government involved, but see whether or not we can be progressive as the chemical strategy and the regulations are implemented. It's promising because there's more dialogue in the UK. There's much more openness, for example, from the HSE or from the Environment Agency to sit down and have some of those discussions at a technical level. And interestingly, we have a roadmap in the UK, or we have a draft roadmap. But for me, it's been quite informative to say that the lead on this has come from the foods area and through the Committee on Toxicity. But there is a draft UK roadmap. I think there's a lot of engagement of scientists. There's discussions going on across the scientists and the regulators. So hopefully this will be picked up and broadened such that it's relevant across the chemical space and DEFRA and becomes a part and parcel of the chemical strategy. So that's what we'd be aiming for as we progress in the UK. It all, of course, comes back to this question around how quickly we can build the confidence of the regulatory community and others in terms of the fact that we are not going to be having unsafe chemicals if we move to NAMs. Now, of course, I'm in completely the other camp as a more investigative toxicologist, that actually when you understand how a chemical interacts with biology, and that being species-relevant biology, being that human biology or being fish biology, then you're going to actually make better decisions on the safety of those chemicals. And if anything, you're probably going to be a little bit more conservative because many of those in vitro systems, as we know, are a little bit more sensitive than sometimes the animal studies are. So, you know, we've got those two schools of thought at the moment, but certainly companies, regulators, we're not going to be in a position of doing anything that's actually going to be more harmful to human health or the environment. So I think the US are leading again here. They've involved some of the European Commission leads in looking at the framework for establishing scientific confidence. And I have to admit this, Michael, but the validation work that we led on in Amden in 1995 or what have you, it's not going to be an approach that's going to be fit for purpose if we want to do some of this at the speed that we're looking for. So it's how do we show that scientific robustness, that scientific relevance, without necessarily what we did in the past to get methods into the OECD test guidelines. That is going to be the need as we go forward. This is going to help, I think. So the US, under the National Academy of Sciences, kicked off an activity last year which was about understanding the variability and reproducibility of the animal data. And they have looked across thousands of chemicals and thousands of animal studies in the US databases. And the US EPA lead, Katie Paul-Friedman, is now starting to talk about the outcome of that. And when you see comments like the qualitative reproducibility of the organ level effects in repeat dose studies of adult animals was 33 to 88%, depending on the grouping. And that figure actually is the one that underpins that. So they've looked at acute, they've looked at chronic and subchronic, so 28-day, 90-day studies and the repro studies. And that's the reproducibility of the animal data for another animal test on the same chemical. And then they were looking at what the benchmark would be for the NAM, the non-animal approach. And what you're seeing is there's a tenfold either way. If your truth was 10 mg per kg body weight in terms of your no observed effect level, then the range of that is 1 to 100. So if your NAM was, or your NAT strategy was predicting between 1 and 100, that is really the realistic expectation of a NAM. So this is going to have to work its way through in terms of how that database then gets brought into US regulatory application. But I think it gives us something we've never had before, which is that understanding of the variability in the animal data. Finally, just the policy piece. There's a lot of new policy measures under the EU's chemical strategy for sustainability. So this came out under the European Green Deal in 2020. Many of them not so relevant for the discussion on non-animal testing, but the one that is actually this promoting safe and sustainable by design chemicals, because I think this is where the NAMs piece actually starts to fit and have a home. And I just want to flag, there's a great figure in this very long technical report from the Joint Research Centre, which starts to look at both new chemicals and existing chemicals, starts to bring in also the classification and labelling and packaging sort of aspects in terms of how you would use NAMs in a tiered approach. And I think, given that that's sitting there in the Commission's own Joint Research Centre report, it's worth thinking about how we build on that in the context of what we're trying to achieve going forward. And Andrew Worth from the JRC actually presented that yesterday, better than I can, but I think that's where the JRC is certainly seeing the next 5 years in terms of their commitments. In the UK, I think we've just got the opportunity to maybe borrow and build on some of the work that's been done at the EU level. So where are we? On this one, whilst we're getting creative in terms of using relevant NAMs, we've still got the whole thing around modernisation of the legal and regulatory requirements, and we still need to develop the frameworks that allow us to actually use NAMs for regulatory purposes. And this should help because the Commission has taken quite a lot of note of the European Citizens Initiative. It had transforming the EU chemicals regulation as one of the 3 aspects of that. And what we're seeing is that is a little bit the backdrop to the commitment that's starting to happen around a roadmap to phase out animal testing for chemicals. Now, they don't intend putting deadlines on that. It's a commitment, I think, to do a plan and to work out what can be done. But I think what we have got is quite a lot of support from the European Parliament. They themselves in 2021 had come out unanimously pretty much in terms of needing a roadmap for phase out. And what we can see is the European Parliament through October have been educating themselves. I was in Strasbourg last month. We were having some very good discussions with some of the MEPs around the status of the science, and it was great to see the alignment across academia, companies, EFSA and the JRC. ECHA were notable, not around the table, but at least you can start to see some commonality in terms of where we are. And then, you know, I think the suggestions that are coming, the proposals that are coming from the NGOs are very strong in terms of the changes that need to happen to REACH. And I think that will be the way that we get the right type of amendments into the REACH legislation. And I think yesterday in Europe, a lot of this was discussed under the Commission industry session at the European Partnership for Alternative Approaches. So, where are we in terms of strengthening animal testing as a last resort? I think there's a few proposals there. One is, you know, why wouldn't we ask for an immediate pause on any animal testing related to cosmetics? Let's have some way, some mechanism, some process where there's much more open dialogue at a European level. You know, we've got some of the training materials there, but there's a hell of a lot of work that still needs to be done in terms of building capability more broadly across scientific and regulatory communities. And we do need to think about service providers. We all know where to go to a CRO if you want an animal study done. How do you know who to go to as your service providers if you want the non-animal assays to be conducted? And then it's all dependent on the incentivization to some extent of having that framework so that everybody has the clarity of what we're aiming at and where those NAMs are going to fit in. So, next to last chart, I am quite passionate that we as scientists do carry on speaking up over the next 12 months to 2 years. I think that's what the tipping point is. I think I am so impressed by the young scientists who come through, who haven't come through an animal background, who understand all of the new methodologies, and I think that's the future, but we need to try to accelerate and make sure the environment into which those younger scientists are coming into is not one that's still fraught with all the battles that we've been having ever since Michael probably started in this space, yeah? So I think that for me is how do we recognise the excellent science? How do we then make sure that that science adds value and can be used for regulatory purposes? we are in a situation of trying to convince people who are understandably quite a considerable way from the science. And how do we best do that in terms of our engagement and communication? So that's a bit of a challenge sometimes for us as scientists. How do we simplify our messages? How do we come across with, not having all the answers, which we don't have, but confident enough in what we do know and how we can best use that? So I do think that if we want our science to have impact, we've got to stay in there. We've got to have the right conversations, and we've got to team up. We've got to team up with those who understand the policy and who are communication specialists. So again, it becomes a bit of a team sport in terms of how we crack this one. And so I think final slide, if we're going to turn our ambition into reality, I know we're here as animal advocates and we understand the importance of the 3 R's to us, but I think for the next 5 years, preferably not the next 15, we need a 3 T's. We need transformation of the chemical regulations. We need transition from using animal data to using non-animal data routinely, and we need translation, which is the ability to be able to interpret that non-animal data for safety decisions, which afford as good as, if not better, protection for human health and the environment. Thank you.